Hello, and thank you for joining us. I am Jasmine Navarez, Digital Content Coordinator at Simulations Plus. I'll be hosting today's webinar behind the scenes, making sure we don't have any technical difficulties. In this webinar, Senior Principal Scientist Jeff Woodhead will introduce you to the new features and enhancements available in RENAsym 2A. Director of Business Development, Andy Mueller, will serve as our moderator today. A few housekeeping items before we begin. We take your privacy rights seriously. By registering for and attending this event or participating in the Q&A session, you are allowing us to contact you for follow-up. You may ask questions via the questions panel on your dashboard at any time. If you need assistance, please use the hand raise icon. We'll address all questions in the Q&A after the presentation. And I'll go ahead and pass it over to Andy to introduce our speaker.
All right. Thank you very much, Jasmine. Thanks to all of our attendees today. Appreciate you carving some time out of your busy schedules. And thank you also to our presenter, Jeff Woodhead. Jeff's going to be a great speaker for you. He's a 14-year veteran of QST modeling and simulation, and he's been our RENAsym development lead for the past 7 years. A little background on RENAsym. RENAsym was originally funded by an SBIR grant from the National Institute of Health, and is currently funded directly through our DILIsym and RENAsym consortium. If that's something you might be interested in joining or participating in, please do let us know. We'd be happy to connect you to that. At present, RENAsym version 2A, which was released this past May, is available to you for licensing. With that brief intro, we'll hand it over to Jeff. Take it away, Jeff. Thank you.
Thank you, Jasmine and Andy, for your kind introductions. And it's time to take you through what's new in RENAsym v2a. This is our standard disclaimer. We're not doctors. Don't treat an individual patient based on simulation results, obviously. And so what I want to talk about today is to kind of give a brief introduction to RENAsym for those of you who aren't necessarily as familiar with it and haven't been around since v1a came out. To kind of talk you through what QST modeling is and what RENAsym does that can help predict whether a compound is going to cause kidney toxicity or not. And then I'll take you through some of the brand new stuff that we're really excited about for v2a that we've just released and are really interested in sharing with y'all. So I'll take you through the introduction first and then start to go over the v2a specific things. So what does QST do? And at its base, at the very simplest level of what QST and what RENAsym is it helps you make decisions on whether or not a compound is going to cause injury and whether or not development can be continued, etc. So the idea here being that you can put a compound into RENAsym, it'll predict whether or not it's going to have an AKI liability, potentially saving you some money on a clinical trial that might fail, allowing you to potentially choose which of a series of candidates might be best for your lead from a perspective of kidney safety, and to be able to enable communications with regulators. For those of you who are familiar with our liver toxicity platform, DILIsym, that has been used in regulatory communications quite often and is pretty well established in each of these particular things. And RENAsym aims to do the same thing, but for the kidney rather than for the liver. So we are Simulations Plus. I'm sure most of you are familiar with Simulations Plus, but we are aiming to be a all-in-one shop for all of your simulation and modeling needs for any particular aspect of drug development and delivery that you have. Simulations Plus can help make that better with our simulations products, whether it's from cheminformatics and you're looking at a compound development or first in human trials, all the way up through communications with medical staff and regulators. So lots to offer here. We've been in the QSP business for a very, very long time, doing a lot of support for several decisions, whether or not it's whether to kill a compound or keep it going, or whether to select this lead or that lead, how to communicate with regulators, etc. We've had a great team that's worked on all of this as well, and over between our liver toxicity and kidney toxicity teams, as well as our efficacy teams. We've done a lot of different compounds, a lot of different disease areas. So there's a lot of experience here that went into developing this model and all of our other models as well. To kind of drive that point home, this is we're focusing today on acute kidney injury, obviously, but this, as I mentioned earlier, we have another QST platform that's focused on liver injury that's been around for now 15 years, I think, and is just quite well accepted and respected in the community, as well as several very well validated and well tested QSP models for a whole host of different disease areas that I'm not going to necessarily sit here and list because it would take me all week. So, getting to specifically what RENAsym and DILIsym are, they're funded by these consortia, which are some 15 years ago or so. We put this together for DILIsym. We're getting the buy-in of various pharmaceutical companies who wanted to be able to reduce the role of drug-induced liver injury over the course of drug development. Started in 2011 and has included 21 different pharmaceutical companies over its lifespan. And we expanded to RENAsym once the SBIR ran out. So we've been able to use this and been able to leverage our consortium partnerships so that we're developing this in a way that best benefits those looking to develop the most safe and effective pharmaceuticals. And QST itself, what it does is we combine our understanding of the biochemistry of the target organ. In this case, we'll talk about the kidney. With our understanding of what are the various mechanisms, what are the things that can go wrong that a drug might cause in the kidney, and then combine that with an estimate of kidney-specific, tissue-specific exposure to predict toxicity and its effects on the cell and then the organ itself. And so we like to do this in a population-based method. So we have simulated populations. We'll call them SimPops. We'll talk about that in a little bit, where we are able to predict the interpatient variability in these toxicity responses and predict whether a more sensitive individual might respond, whereas a normal, healthy individual might not. RENAsym itself has human and rat models. There's also a limited mouse model as well. We have actually expanded RENAsym to from the proximal tubule, where we really concentrated our efforts in V1A. We've included models of the glomerulus and of the distal tubule as experimental models. We focus on the cellular level in and of itself, but then both drill down and into the biochemistry so that we understand how the drug itself is really affecting that biochemistry, whether it's through oxidative stress or mitochondrial dysfunction or some things we'll get to talk about here in a couple of slides. And then going, zooming out into looking at things like the systemic inflammation that would be caused by injury, release of biomarkers, and the renal function, the effect that has on the system. So we've have quite a few exemplar drugs listed here, and going forward, we hope to add even more to that list. The main idea of RENAsym is that we have these dynamically interacting submodels that are one focused on cellular injury. So what's the drug doing in the cell, and how does it affect ATP balance and cell death within the cell? And then that interacts with the renal pathophysiology. So as these cells die, what is that doing to the kidney, and what is the kidney health doing to the rest of the body? Um, and so starting on the renal, the broader tissue portion of things, there's this physiology submodel that we've based on a model that's wound its way through the literature, most notably an extension of a model that Melissa Hallow and her collaborators have developed, that we've kind of added pieces to and developed beyond where it was then. So, that model includes a lot of the renal vasculature, and so it's sort of the effects of the RAS system and those sorts of things. We've added some pieces on nephron specific. What does it look like to kill a nephron? How do nephrons die? How do nephrons survive? And those sorts of things. So, kind of tacking pieces on to some good work that's been done by other people in the field as well. Within the cellular model, we include a model of mitochondrial activity. This is a model that was taken a lot from the DILIsym case, where you have a fairly well regarded and well developed mitochondrial model within the liver, and we took it and we adapted it to what the kidney looks like. The ATP usage is different. Substrate usage is different. There isn't the Randall cycle that occurs in the liver. It doesn't occur as much in the kidney. But the toxicity mechanisms still happen, right? You can still have the electron transport chain uncoupled. You can still, pardon me, you can still have the electron transport chain inhibited. You can still have the proton gradient uncoupled. You can still affect the mitochondria in those ways, and that will still lead to ATP decline and cell death. We pulled over the idea of oxidative stress generation also. A lot of this was explored and done in DILIsym, and then adapted to the special case of proximal tubule cells in the kidney and later podocytes. And of course, there are in vitro assays that can determine how much drug is contributing to ROS, and then what we know about kidney biochemistry will dictate how much oxidative stress can either lead down the road to apoptosis, or if ATP depletion gets too severe, it heads towards necrosis. Back out of the cell, once cells start dying, one of the things that people look for in the clinic, and those of you who have dealt with compounds failing in the clinic know this, is that you look at certain biomarkers of the kidney, and if those start to, there's certain ways in which they'll start blinking, flashing red lights, and that you'll know you have a problem. And one of the key ones is glomerular filtration rate. So obviously we had to have a model of GFR in RENAsym, which we've included that in the as in something of an add-on to the kidney model that we talked about earlier, where there's filtration pressure from the glomerulus into the proximal tubule, and so that and serum creatinine is also determined as well. We added a couple of urinary specific biomarkers that are kind of emerging. These two are a little bit more established. Alpha GST, which is shed from after dead PTCs release their contents. Alpha GST ends up getting into the kidney tubule lumen and then secreted into the urine. Same with KIM1, which is a newer biomarker where it's actually shed a little bit before there's toxicity during the dedifferentiation process of recovery. So certain sublethal injury can still be picked up using KIM1, and we have that in the model as well. There's a pretty substantial inflammation model. Once cells die, they release these DAMPs, these damage-associated molecular patterns that lead to inflammation, and inflammation obviously can contribute to both injury and repair depending on which cytokines and which cell types predominate. And so we have that. That's also quite heavily informed by our work in the liver, as well as our extensive work on inflammation in other places as well. And as I alluded to earlier, we like to have this in a SymPops format where we have some variability in kidney biochemistry. The V1A SymPops originally had several basic biological parameters that varied, such as body weight, sodium intake, etc. And we were able to take those, vary them, generally compare those to clinical data to make sure that we have a reasonable distribution, and whether, in this case, we'll look at, say, crystal formation and making sure that we have the right amount of patients in there. And the idea of how to use RENAsym is you take some in vitro mechanistic data that talk about how much a drug could possibly affect this particular pathway, whether it's oxidative stress, whether it's mitochondrial toxicity. Then you can pull in some physicochemical properties depending on how soluble or insoluble you expect your compound to be. So we combine that with PBPK modeling that gives us exposure both in kidney tissue and in the kidney tubule lumen, as well as kind of an understanding of what our target population looks like and what our potential dosing protocols look like. All of that is combined to give us predictions that will help predict whether or not a compound will potentially cause injury and what to do about it. So that's the basic overview of what RENAsym is. Now I want to take us through a few of these new features that have come out in RENAsym v2a. RENAsym v2a was released earlier this year, and it has quite a few new features. And I did want to point out that as consortium members, the companies that contribute to the RENAsym consortium get to drive the bus in terms of what things we add to each successive version. So, for example, the consortium suggested that biomarkers, and especially certain novel biomarkers that were under investigation by the Predictive Safety Testing Consortium, or PSTC, under the aegis of the FDA, were interesting to include in RENAsym. So we did so. Members wanted us to look at podocytes and distal tubules, and so we included representations of both of those in there. As we go forward, there will be a RENAsym v3a that will come out eventually, and the members were able to give us their insight and tell us what they thought would be most useful, and that'll drive what comes out for the next version of RENAsym, and so on. So joining this consortium gets you a seat at the table and gets you can see what things you want to come out of RENAsym. If you come out of this webinar thinking, oh, well, RENAsym sounds great, but I wish it had this, then the best way to make sure that it potentially includes that is to join the consortium so that you have your voice heard on what's most useful to you about drug-induced kidney injury. So let's talk biomarkers. Since RENAsym V1A, we've included quite a few tubular AKI biomarkers. There's quite a few that we've talked about: NGAL, NAG, clusterin, osteopontin, and we like to say these are all somewhat works in progress because we understand some of these scientifically, but the data on the ground are a little bit thin. And so, I'll talk about one of the specific representations here and how that affects things. But there's some new data that came out that we've been aware of since RENAsym V2A development ceased, that we will, are trying to start to include potentially in the next version of RENAsym and going forward. So each of these are they're experimental biomarkers, and so they're experimental models, but they're important to include because of how many companies are starting to pull these in-house and use them as potential early predictors, more sensitive predictors of kidney injury than the standard serum creatinine, GFR, yada yada. So these are the new ones: NGAL, NAG, clusterin, and osteopontin that were included in V2A. Alpha GST and KIM1 were included in V1A, and of course are still there. So just as an example, I wanted to take y'all through the process of modeling NGAL, which is neutrophil gelatinase associated lipocalin. We'll just call it NGAL. It has quite a few different potential functions in the kidney, but the big thing about it is that if the kidney starts to be injured, that its expression goes up, whether it's as a recovery thing or an injury signal or what have you. It goes up, and it can increase in the plasma for a number of different reasons, uh, whether it's infection or inflammation, injury, etc. But the the main point is that it very rarely shows up in the urine unless the really efficient reabsorption of NGAL is impaired in the proximal tubule. And so urinary NGAL ends up being a pretty good predictor of AKI, um, and some sort of proximal tubule cell functional loss. And so that's what we are intending to represent in RENAsym. First, you want to look at kind of some of the data that are out there. You see kind of some of these co-localizations with ATP decrement. You can see that as ATP is decreasing, the NGAL expression is increasing. These are largely animal models, but we can extrapolate that to the other animal and human case. So using some of these data, we can develop a model that leads to a representation of NGAL and a mechanistic representation of NGAL within the kidney. And what we're able to put together is that there's using some of that data from the last slide, as well as potentially some data from actual injury cases where NGAL is measured. We understand that there's a functional link between ATP loss in the proximal tubule and the production of NGAL. In this case, we developed this model before we put the proximal tubule stuff in there, and didn't really gain us a whole lot to move NGAL to the proximal tubule for 2A. Potentially for 3A, this is another thing to revisit, right? That looking at NGAL is released by the proximal tubule into the plasma, but we see it released into the plasma from a production signal currently. It may or may not help us, honestly, to include it in the to go deeper than this. One of the things you do as a modeler, you look at how simple do I need to, how simple or how complex do I need to make the model to get us to where we're trying to get to. And as we'll show in a couple of slides, this ends up being a pretty good model. And so, it goes from the plasma into the lumen, into the bladder, and out into the urine. And as I alluded to, there are some data out there looking at NGAL in cisplatin nephrotoxicity in humans, and we are able to recapitulate those data pretty darn well. Looking at NGAL production signal, just mechanistically speaking, we were able to get it to line up with the time course of the injury. Cisplatin is probably our most validated compound, both mechanistically and in terms of other biomarkers. So we're pretty confident that the level of injury is appropriate here. And so we were able to calibrate the level of injury with the level of NGAL release. That worked out well for us, but there's always the issue of data poorness. And so this is largely the only data set that was out there at the time of V2A release. As I mentioned earlier, the PSTC has put out their data now, and that'll help us a lot going forward with these. But experimental biomarkers, experimental models, and this is still something that's pretty useful to a company that's looking at some of these novel biomarkers for potentially predicting a novel compound's potential to cause AKI. I want to talk a little bit about some of the other submodels that we've included in RENAsym V2A. This is the podocyte life cycle. And the podocytes have a somewhat unique life cycle in terms of the cells that we've looked at in our QST models. There's this somewhat progenitor population, these PECs, these kind of epithelial cells that mature into podocytes that themselves don't really undergo mitosis. In fact, if a podocyte undergoes mitosis, that's a bad thing. It's called mitotic catastrophe, and they often die of it. And so as podocytes die, they're then replaced by this progenitor population. And so there's a way of getting that, calibrating it to a steady state thing. And then as podocytes are stressed and potentially killed, that kind of deranges this whole process. And that's kind of the center of our podocyte toxicity model now, is that how podocyte injury affects this cell cycle development. And so we have this several ideas of how the glomerular injury is caused by a drug and indeed by other things as well. There's also the once podocytes are dying, once the podocyte population decreases, how does that lead to kidney functionality? So there's a lot of good data out there on percentage of podocytes, especially in rat models that link percentage of lost podocytes to percentage of normal filtration that you possess. And it seems like you can lose about 10 to 40% of your podocytes without a lot of functional consequences, and that the real injury occurs after that point. And you can see something similar in humans as well, where there's a general fall off of GFR after a certain point. So we were able to calibrate that within our model so that podocyte injury will lead directly to a loss in kidney function after a certain threshold. Also including the distal convoluted tubule as well. And it has an option of both ROS and mitochondrial toxicity, just as the podocytes and the proximal tubules do as well. What we do lack here are compounds, and so we call this something of a experimental model at the moment, because we haven't looked at its actual effect on a compound yet and calibrated some of those subcellular processes to those data yet. One thing that I have neglected to talk about so far, but that's an important portion of RENAsym, is the crystal nephropathy model. There's a crystal nephropathy model in V1A And we've expanded that significantly for V2A. And we've expanded it in two ways. One is that we've included crystal formation in the distal tubule. And the main thing is that we've made the mass balance make a little bit more sense by kind of shifting up how data come from GastroPlus and your PBPK program into RENAsym itself, so that it's the crystallized and non-crystallized and dissolved and non-dissolved are keeping something of a mass balance in there, and so that we can apportion out the compound into each of those compartments as crystallization happens in real time. That's something we didn't necessarily have in V1A. We had more of an approximation model. In V2A, we're making that a lot more explicit, and so it's a lot more biologically relevant in terms of how it's apportioned out within the kidney tubule. So that's been improved, and that is a key part of RENAsym, because there are a decent amount of drugs that do cause crystal nephropathy and that people are interested in predicting. I'll get to a couple of those predictions in a little bit, but I want to talk about the new SymPops as well. As I'd mentioned earlier, the V1A largely included some biological constant variability. We've added glomerular volume for the podocyte model into the SymPops, but we've also added variability in mitochondrial dysfunction. And we had something of a back and forth on how much variability to include in mitochondrial dysfunction. Fortunately, glomerular volume, that was pretty well calculated. There's quite a few papers that have calculated glomerulus and podocytes per glomerulus and variability within that, and so that was pretty straightforward, and our team did a really great job of putting that together. OCR wise, we looked at some of the OCR measurements in after FCCP administrations in RP-Tech cells, looking at how variable the respiratory reserve scalar was. This is kind of the same approach that was used in DILIsym, and we were able to measure kind of what the variability in respiratory reserve might be after a toxic insult. So that was able to be incorporated into the model as well. And before I open things up for questions, I wanted to go through and give a couple of examples of compounds, since that's what we're really looking for here, is can we predict injury in injury compounds? And so we'll look at a couple of our new Kook compounds. We've added polymyxin B, lithium, and doxorubicin. Polymyxin B and doxorubicin are positive controls. Lithium is an interesting one because it gets linked to kidney injury a lot, but the evidence for it is incredibly weak. And so we kind of included it as a negative control at the moment, but some of the clinical toxicity data are so difficult to deconvolute from other things that are going on in individuals that have been on lithium for 3 or 4 years. So we'll obviously going to come back and fully re-examine that as time goes by. Polymyxin B, we parameterized both OCR and ETC inhibition and oxidative stress. You saw both in the clinic. The OCR curve was a little bit funny looking, but we were able to parameterize it anyway, and getting to doing things kind of in the standard way we do things. This was something of a pure prediction, right? We didn't calibrate the model at all to polymyxin B. It was a lot of the calibration work was done with cisplatin in some of our earlier compounds. This was something that we just kind of took right out of the box and put through our assays and fit them the way we would normally fit them during a prediction. And we were able to correctly identify polymyxin B as a kidney toxin. Now it's introducing some really high levels of kidney toxicity here with an entire population's GFR declining pretty rapidly. PMB is a pretty serious kidney tox problem, but that's normally 20 to 40% of the population, and this is the entire population. So, obviously we're over predicting a little bit, but I think in terms of is it a yes no, we are correct in that regard, right? We got the yes no right. We got the yes right. We have paper coming out that will hopefully be coming out on this soon. Celeste Vallejo will be our first author on that. So keep your eyes peeled for that in the coming months. So it'll be a nice publication for us. Doxorubicin as well, somewhat similar story. Again, using some toxicity assays, but in this case, there was less in vitro toxicity assays. And this is actually an interesting one because, we talked about the in vitro assays as being the key inputs, and they really are. They're the gold standard. But if you don't have those and you have some other data that potentially could be used to suggest that there's oxidative stress or to suggest that there's ETC inhibition. In this case, with doxorubicin, in the literature, we had rat in vivo oxidative stress, and we had in vitro cardiomyocyte data, which we're not necessarily used to using. So we went back and forth on parameterizing that a few times. And while again we overpredict AKI, it still gets the yes no right. It's yes, right? It's doxorubicin is a kidney toxin. So that was a good thing. We do know there is some sensitivity because we reduced the exposure by an order of magnitude and got no signals. So we know that there's sensitivity there. It's not just automatically predicting yes for every compound. And we do have quite a few other negative controls. As I mentioned, lithium we predict as a negative control, and some other negative controls, for example, acetaminophen within the kidney space as well. Moving on to the crystals, this is looking at this new crystal nephropathy model that we've included in V2A. Indinavir is our main exemplar here. We are able to predict both the effects of crystals and a fairly broad range of crystal aggregate size for indinavir within the kidney here. So that was nice, and they're able to predict some serum creatinine increases like you see in the clinic with indinavir. So that was something that's new in V2A. We didn't get the serum creatinine increases before, and we do now. So again, qualitatively correct and getting closer to quantitatively correct as well here with indinavir. Wanted to add on that two other compounds that cause crystal nephropathy are acyclovir and sulfadiazine. We get some serum creatinine increases for both of those as well, and that was nice to see, and that cohered a lot with clinical experience for both of those compounds. And so I'll bring things to an end here. We've got some time left for question and answer. I wanted to leave you all time to pepper me with questions about RENAsym and about what it can do, what it can't do. Where are we with with being able to predict AKI, and how we can help you as a company de-risk certain compounds, choose your compounds better, and communicate with regulators about kidney injury. So again, I think if Jasmine mentioned how to put questions in, and I'll see if we have any.
Cool, thank you. So again, feel free to add questions to the chat or in the Q&A. Bizwatchit has a hand up. Can we unmute? Let's see here.
Don't know, can we?
Let me try this.
Oh, yeah, he's unmuted now. Oh, I was muted again.
Let's see here. Jasmine, can you help with an unmute?
Yeah.
Let's see here.
Oh, there
Hello,
we go.
sir.
Yes,
There we
hi.
go. Go ahead.
Yes, sir. Myself Vishwajit. I am from India.
Great.
Yes, sir. Sir, I want to a section for gastrofloss, how to use gastrofloss and how to build a project in gastrofloss. I want to see that,
Right.
please.
So yeah, so GastroPlus PPK model. Yeah, obviously our softwares are same company. We're meant to have GastroPlus feed into RENAsym and have the two really work together. GastroPlus model. If you wanted to build that, there's a number of different ways to build that, and a lot of other things to
Sir, and
ways
also
to
I
learn.
want to know that, what the parameter, what the physical chemical parameter we have to find out for the using the gastrofloss for a development of a new medicine.
So the great thing about GastroPlus is you can actually have a model of it at pretty much any stage and any level of knowledge you have. The if you just have a structure, GastroPlus has an ADMET predictor module that can predict some of those physical chemical properties for you. If you've measured some things, you've gotten far enough along where you have the compound, you've measured log P, you've measured things like the solubility, you've measured blood to plasma ratio, you've measured protein binding. Just some of the standard stuff you have in the your pHs, your pKa's, uh, those sorts of things. All of those can be used as inputs into GastroPlus, and then the next layer is metabolism and transporters and all of those things. So you can really put a model together with any level of data that you have, and then using that predict. And GastroPlus is able to predict both kidney tissue and kidney tubule concentrations. So you're able to take those and then bring those into RENAsym to predict your kidney toxicity. So really, realistically, you don't have to be at a certain point in development to be able to use GastroPlus and hence RENAsym. You really just have to have a compound.
Sir, hello, sir.
Yes.
Sir, I want to know about that what the gastroplogy uses in pharma industry. Actually, sir, I am from pharma background, so I want to know that what is the uses of gastroplogy in pharma industry for the development of a drug.
Pretty broad. The GastroPlus is pretty well used throughout the industry. I don't know exactly how many clients we have. I think I'd have to ask someone over on the PK side about that, but it's a very substantial list and it's pretty broadly used. So I think most companies you end up at will probably have a GastroPlus license or two to be able to use in conjunction with this.
Yeah, thank you,
Um,
Jeff.
Thank you,
Got
sir.
kind of a comment here, sort of building off of what our participant was speaking about. So when expanding capabilities of in silico modeling, model-informed drug development, including, PBPK modeling, QST, QSP modeling overall, obviously real-world relevance is important take into consideration and understand, so that we're, informing our models and databases for application and current use case scenarios. Where are we in terms of moving beyond, the traditional healthy volunteer populations into populations like chronic kidney disease, type 2 diabetes, those sorts of things within the context of RENAsym?
Yeah, that's an excellent question, and it's one that we've definitely thought about a lot. And as we develop forward through V3A, this is actually an interesting point to make, because when I talked about the consortium and the consortium's effects on development, and being a part of the consortium, getting you a part of the getting you a seat at the table and getting you to help drive the bus with RENAsym development. This is something that over the course of the last year or so, pharma companies have become really interested in. Okay, great, you can predict NHVs. We're really seeing a lot more problems with individuals with CKD. And CKD is growing in incidence. Can you help us do that? And so for V3A, that's really going to be a key portion of our model development going forward, is can we put together a population that represents chronic kidney disease individuals, and then can we predict whether a compound that may look safe in an NHV population would not be safe in a CKD population, or a population with T2D that hasn't quite progressed to kidney disease yet. All of those things are things that we're definitely thinking about and looking at moving forward with RENAsym, and that people who are a part of the consortium, and even those who are licensees, will be able to learn about and see as we go forward.
Cool. Excellent. Thanks, Jeff. Not seeing anything in the chat and not seeing any open questions in the Q&A, or any hands raised. So that might take us to the end of our presentation today. Jasmine, can I return the reins to you, please?
Sure. So I just want to thank everyone. For any questions that do come up, our BD team will follow up with you after the webinar. We invite you to visit our website to learn more about RENAsym 2A and our other software platforms. You can also follow us on social media. I put these links in the chat. This webinar has been recorded for playback and will be available on our website and YouTube channel. This concludes our webinar for today. Thank you, everyone.
Thank you.